0.2% of all adult malignancies. And so when you're sitting in a waiting room, or a patient sits in a waiting room, I mean, the person next to you, unless you're in a sarcoma clinic, the person next to you doesn't have a sarcoma. And the person next to them doesn't have a sarcoma. And you're more likely to meet people with breast cancer, colon cancer, lung cancer than you ever are to come across someone with a sarcoma. Usually what we think that the majority of the sarcoma pie is taken by a liposarcoma, an undifferentiated pleomorphic sarcoma, or a leiomyosarcoma, and those three make up most of what we see, and then everything else makes a little bit of a spattering. Okay, if we look overall, and when we talk about the progress made in cancer, we talk about 5-year survival. That's usually what people talk about maybe 10, 15 years ago. Everyone always says, what's my 5-year survival? What's my 5-year survival? Somewhere along the line, that 5-year became this number that we all hold on to. It's that holy grail. And whether or not it's an important number or not is questionable, because certainly you want to know what's my 10-year survival, and you want to know what your 15-year survival is, but everybody holds on to this number of the 5-year survival. And so we see that the 5-year combined cancer survival rates have improved over time. And so if we think that they were about 49% in the 1970s, they're about 69% between 2013 to 2019, and that's for all cancers. And the progress that was made in between the 70s and 2013 to 2019 is likely a reflection of the smoking and decreased number of smoking, but also the introduction of mammography, the introduction of colonoscopies. Unfortunately, when we look at sarcomas, we see that the number of sarcomas and new cases have climbed over the years, but we slowly see an increase in the red line, which represents the mortality. And so where we've been able to improve overall, in sarcomas we perhaps are not improving at the same rate. And there are several factors that play into this. And so with the use of maybe more radiation therapy, radiation could cause a sarcoma. So there's other reasons why people develop sarcomas, and so probably some of the stuff that we end up treating the primary cancer with puts patients at increased risk. There are other issues that come into play, but certainly we have not seen a decrease as we've seen with other diseases, and so it just means that we need to continue to work and continue to make changes. So when we look at the majority of sarcoma trials that we've focused on over the last, since about 2014, so the last decade, so a lot of you may have been involved in some of these trials, and so for many, many years we looked at could we improve upon the treatment of patients, and what we did is we lumped everyone together. So we didn't put a leiomyosarcoma in, we put a leiomyosarcoma and a liposarcoma and an angiosarcoma, and we just put everybody into the same basket, thinking that all sarcomas are the same, and not really taking into account that each sarcoma is unique and different. And when we lump everything together, what we end up doing is not proving anything, because we're not getting enough patients with each type. And so essentially we ended up showing that doxorubicin is a great drug. Doxorubicin is also adriamycin, the red drug. And so doxorubicin is a great drug, and what we've always proven is that it's better than anything else we've tried to combine it with. And so the year is on the left, the control arm is always the doxorubicin. It's either given alone or with placebo, which is essentially giving it alone. And then you get the experimental arm right in the middle, the number of patients exposed to these treatments. And so we're looking at 2 and 4 and 2 and 6 and 5. So we're looking at several thousand patients. And at the end of the day, we haven't developed any new drugs in this space. And the space here is soft tissue sarcoma. The space isn't a specific sarcoma. And so this one is important, this last one, because olaratumab, which is the last thing on the line, olaratumab actually came to market. And so there are patients who received olaratumab. It used to be called Lartruvo. It was on the market for a few years, and it was on the condition from the FDA that Eli Lilly do a confirmatory study to prove that the drug was indeed beneficial, and ultimately it proved that the drug was equivalent. And so the drug was pulled from the market because of that. So now when we do specific trials for specific histologies, this is when we start to succeed. And so when we stop with the assumption that all sarcomas are the same and start with the assumption that each sarcoma is very unique, we start to succeed. And so we have in 2015 the introduction and approval by the FDA of trabectedin. which was a drug that's been on the market around the world, and a lot of American patients had to go either to Mexico or Canada over the years to get it before 2015. But then it was tried in the US with a better study, and the FDA approved it, and it is very readily used in both leiomyosarcoma and liposarcoma. We have eribulin, which was FDA approved in January of 2016. Similar trial in liposarcoma and leiomyosarcoma. The FDA approval comes for liposarcoma, but most of us in the world of sarcoma do use this for leiomyosarcoma as well. We have pexidartinib, that's approved for tenosynovial giant cell tumor. Tazemetostat got approved for epithelioid sarcoma. Nab-sirolimus for picomas. Atezolizumab, which is the first immunotherapy that's approved for alveolar soft part sarcomas. And nirogacestat, approved for desmoid tumors. And then there's Tecelra, which just got an approval for synovial sarcoma in August of this year. And so we are making headway, and the headway is made because we're actually realizing that each sarcoma is different. And so here is an example of Tecelra. So this is otherwise known as afami-trezgene autoleucel, or afami-cel is the other name. And so what we've done here is we've engineered T cells, which are the body of natural T cells to fight things. And so there are engineered T cells, so they're engineered out of the body, and they target a certain receptor called NY-ESO-1. And we know that NY-ESO-1 is a target for patients who have a synovial sarcoma or a myxoid round cell liposarcoma. There is an antigen called MEG-A4, which is expressed at varying levels in multiple solid tumors, but is highly expressed in synovial sarcoma, and it's expressed with an HLA, and so HLA is in a blood test. And so this is only for a certain number of patients who have the right HLA. And so afami-cel is an HLA T cell therapy that is created out of the body, and it targets MEG-A4. And so what this study did is it looked to include patients who had a metastatic synovial sarcoma or a myxoid round cell liposarcoma. They had prior doxorubicin, prior ifosfamide, and they essentially had to be very functional. And they had to have a certain blood type, a certain HLA type, because the MEG-A4 gets expressed with this HLA. And so if you met the inclusion criteria and you didn't have an exclusion criteria, then essentially you were able to undergo treatment with this. And so essentially what happens is you end up getting blood removed, which is leukapheresis, which is the second box. And so that is a collection of the patient's own blood in order for a product to be made from the patient. And then what happens is you end up getting chemotherapy, which is the lymphodepletion part. And so under lymphodepletion, patients are brought into the hospital. They're given several days of chemotherapy in order to almost wipe out their bone marrow, bring down all the counts. And then essentially the product that was created from the patient's blood is reinfused back into the patient. And that was for 44 patients. And what you see is, oftentimes you'll see these type of waterfall plots. So essentially the graph on the top, each line represents a single patient. The middle line here says 0. So that's the starting point where everybody starts a trial. If your tumor gets worse and grows, the line goes up. If your tumor gets better, the line goes down. And if the tumor stays the same, the line sort of stays. We use these arbitrary or not arbitrary, we use these definitions. That stable disease, so if you walk in your clinical trial and someone says you have stable disease, that means your tumor has not grown by more than 20%, and that means your tumor has shrunk, but not more than 30%. And so if your tumor shrinks by 29%, it's considered stable. If it grows by 19%, it's considered stable, but usually it's going to fall close to 0. And so what we see is the majority of patients, the lines go down, meaning the tumor shrinks. And so, and then anybody who falls below this dotted line, these are the patients that we call responders. And so certainly when you ask what is the likelihood of responding to a trial and research, you have to understand that if your tumor shrinks by 29%, every human being is going to say my tumor is responding, but our definition of response gets triggered at 30. And so what you really want to know is what is the likelihood of the tumor getting smaller. And so where responders only fall to these, all of these patients are really responders as well. They're just not responding as well. And so in this trial, the response rate is about 37%. So 37% of patients have their tumor shrink by more than 30%. For synovial-sarcoma, it was 39%. Patients get a cytokine release syndrome, which is like a flu-like syndrome, which happens when you infuse T-cell therapies. And really the conclusion was that this is a highly effective therapy, and so a family cell treatment resulted in a durable response in a heavily pretreated patients with HLA-02 and NEG4 expressing synovial sarcoma, and the drug gets approved in August 1st. So this is a very targeted treatment. So what are the downsides of this? The downsides is the majority of patients don't have HLA-A02, and so the majority of patients aren't eligible for this treatment. And then the question is, could we replicate this success in leiomyosarcoma? And so a lot of what we do in other sarcomas, we have to figure out, could we tweak it in order to make it as effective for patients with leiomyosarcoma? Because it's great if it's helping the synovial sarcomas, but that's a small part of the entire pie. And what we want to do is increase accessibility for everyone. So when we talk about research and advances in leiomyosarcoma, I sort of broke this into several boxes, and some of it you've discussed today, and I don't think I'll touch on it, and others I will. And so one is talking about genetic research, and so this is the investigation of mutations in the leiomyosarcoma cells. One is looking at new biomarkers, so things that could be targeted, that if you target it, you end up having a positive response and the tumors aren't turned off. We'll look at some ongoing clinical trials that are focusing on new therapies, combination therapies, or personalized therapies. Immunotherapy, I think Dr. Chmielewski was here earlier that discussed a lot of what is being done in other cancers, and could we bring this into leiomyosarcoma? And really a lot of this is about precision medicine, tailoring treatments, plans based on genetic testing of the tumors, and it's really making a very conscious understanding that the genetics of your tumors are not the genetics that were passed down from your parents. The tumors have their own genetic profile, and so it's not seeing a cancer geneticist who does the genetics from your parents, it's really looking at the tumors themselves. And so if we look at investigating mutations, so why do we study mutations in an LMS cell line? And so what we know is mutations drive tumor growth and affect how LMS responds to treatment. So I always use the analogy of a car. People drive places. If something happens to the passenger in the car happens to the car keeps driving. If something happens to the driver can't continue. And so what becomes very important is to understand what is the driver versus what is the driver versus what is the passenger. And we don't want to give drugs that will affect a passenger, we want to give drugs that will affect the driver. Okay, so what we want to do. Are we okay?
What's wrong? She's lost her purse.
Is it reimbursed?
This one was missing. Should we do it in the break? In the break, please. Sorry about that.
Okay, that's fine. Okay, so what we also want to understand is how the genetic makeup helps identify potential treatment targets. And then we want to also look at the common mutations found in LMS, but that doesn't make us forget that there are some very rare ones, and that if everyone in this room had LMS, you all are not the same. You all don't have the same genetic address. And so the question is, how do we hone in even deeper on the genetic address? And when we have genetic trials, there are patients that I've treated over the years. I had one a few years ago that we treated like a melanoma because their genetic profile was much more in keeping with a melanoma than a sarcoma. And we had someone that was treated like a rare form of lung cancer because their genetic profile looked identical. And so it's what we would start to look at is some agnostic, tissue agnostic treatments, meaning you don't care about what the tissue says, you care about the genetics instead. And so the most common genetic mutations, if everyone in this room had a leiomyosarcoma and everybody had a genetic test done, and the common genetic tests are done by like Guardant and Tempus and Caris and Foundation Medicine, or in-house testing at a university. Most people will have a TP53 mutation. So TP53 is the most common mutation found in cancers. It's over 50% of leiomyosarcomas. It plays a role in DNA repair and cell growth, and so you can't repair DNA and you can't repair cell growth if your TP53 is mutated. And so it's what we would call as a tumor suppressor gene. So when it works, it suppresses tumors. When it doesn't work, which is most people with leiomyosarcoma, the tumors can't be suppressed. And so that is a great target. Many, many years people are looking to target TP53. It's a very hard target to find. And there has been recent progress with some work, which I'll show you in a few minutes. RB1 is another one that's associated with tumor suppression, disruption, promotes uncontrolled cell growth. And so a lot of patients have an RB1 mutation. And so that mutation produces uncontrolled cell growth. And there are some pathways where we think we can make by targeting the RB1. A lot of patients also have MED12 mutations, which is often in uterine leiomyosarcoma that is linked to the smooth muscle tumors, but not in other leiomyosarcomas. So when we look at the mutations, how do we do the research? The research that we're trying to do is trying to create targeted therapies. So these are drugs designed to block specific mutations in the leiomyosarcoma cells, which is a personalized treatment. We're trying to do biomarker development, which is identify genetic markers that predict how a patient will respond to certain treatments. And then the clinical trial is the way we test these therapies, which hopefully will provide access to patients, all patients, to drugs. So the goal of a clinical trial is a little bit counterintuitive. The goal is not to give access to patients. The goal is really to get a drug to market. And so when you're doing a clinical trial, which is sometimes counterintuitive, you're trying to get the best patients, because the best patients tend to be most compliant. They get through their therapies, they're more likely to succeed on the therapies, and you're able to see things with a lot of clarity. If you're taking patients that have gone through too many lines of therapy and this is their last hope, that's not what you want for a clinical trial. A clinical trial is best used as early as possible when the patient is as fit as possible and able to get through all the treatments so that you could really see if the treatment works. You don't want anything confounding, so you don't want a patient to be too sick, and next thing you know, they shouldn't have been put on the trial, and next thing you know, they don't comply with the trial, and then a drug never comes to market because the wrong patients are put on. And so you really want to stack it with the right patients so that everybody gets access if it comes to market. And so examples of some research currently being done in leiomyosarcoma as well as other tumors is going to look at T-cell therapy. So this is the same therapy that we talked about in the synovial sarcoma, where there is a drug that has come to market. And so this is an adoptive cell therapy that involves genetically engineered a patient's own T-cells. So as I said, you remove cells from the body and re-engineer them, and these will attack a specific thing. And so in the context of P53 mutations, which are the most common, there is TCR therapies that are now being done for certain P53 mutations. And so P53 is a crucial tumor suppressor gene that regulates cell growth and cell death. Apoptosis means cell death. And when it's mutated, it loses the ability to regulate everything. And so then it just becomes uncontrolled. And so a mutated P53 is considered a neoantigen, meaning it's recognized as non-self. And so P53 is considered, everyone has P53 because this is a normal process that we all have so that we all are not forming cancer every time a DNA doesn't get repaired properly. So P53 suppresses that DNA from replicating. And when it mutates, the body can recognize this as different because it's not normal. And if the body could recognize it as different, we could create a process by which we could attack it. And that's the goal of T-cell therapy. And so TCR therapies could target mutant P53 peptides that are present on the surface of cancer cells. They're presented by an HLA molecule, and so everyone has HLAs, and that's what you're born with. You engineer them out of the body to specifically bind to this mutated P53. So when they get reinfused in the body, they go and attack P53. And so there's certain hotspot mutations on P53. And so if you have, let's say you have a NGS report from Tempus or Guardant or Caris, the one you're looking for is the R175H, the R248Q, or the R273H, which are found across many cancer sites, but there is currently research being done looking at TCR therapies for these. And so what you're hoping for is that you can replicate the work that we've seen in synovial sarcoma. And so the study that specifically is looking at it is from Neogene Therapeutics, and it is a study of NT175 in adult subjects with unresectable, advanced, or metastatic solid tumors that are positive for HLA, O201, and TP53 R175H mutations. And so this represents almost identically the work that's currently being done in synovial sarcoma. But TP53 is a very common abnormality in leiomyosarcoma. And so you would hope that while this may only affect a very small percent, it is still headway for some patients. And unless you do genetic testing, you can never find these things out. Because this isn't from a pathology report. And that's the other reason we need to make sure that every patient has genetic testing of their tumors. So this was the original slide looking at the current research and advances in leiomyosarcoma. We've discussed the role of genetic research. So now let's focus on new drug development, combination therapy, and personalized medicine, looking at clinical trials. And there are results that have recently come out. And so here is a New England Journal of Medicine paper, which is probably the most esteemed journal we have. This is from September 5th of 2024. So this is looking back just 2 months ago. And this is in leiomyosarcoma, and it is the combination of doxorubicin with trabectedin, with a trabectedin maintenance therapy. So we don't really have a lot of maintenance therapies in this disease. And essentially what happened is patients were given 6 cycles of doxorubicin with trabectedin, followed by 17 cycles of trabectedin alone, versus 6 cycles of doxorubicin by itself. That's the top bars. And so we had 74 patients got the combination, 76 patients got doxorubicin alone. If you want to know who these patients are patients with metastatic disease or unresectable disease who have never had any chemotherapy, and so this would be a first-line chemotherapy. These patients have not had doxorubicin, so this resets the standard of care. And so essentially what you see in the second graph here, so this is looking at percent of patients, so this is 100% all patients, and this is months since they are randomized, and looking about how long these patients are living. And so the top curve is looking at, the top curve is the combination of the 2 drugs, the bottom curve is looking at doxorubicin by itself, and what you clearly see is that the top line is doing better. What you also like to see from a statistical standpoint is the lines shouldn't cross. The moment they cross, then there's a likelihood that this is by chance alone, or that there is no difference. When they never cross, it means one is superior. And so doxorubicin and trabectedin is superior to doxorubicin alone. And so what we see is that amongst patients with metastatic or surgically unresectable uterine or soft tissue leiomyosarcoma, first-line treatment with a doxorubicin trabectedin therapy, followed by a maintenance therapy, would now be considered first-line standard of care. And so if someone's treated today, this is how they should be treated. If they got treated 6 months ago, well, in all likelihood, they didn't get this. And so this is just showing that there is an evolution to the treatments that we have, and where things were standard at one point, as soon as you get more information, then the standard of care changes. And it takes a little while for things to catch up. The sarcoma community catches up much quicker, but not everyone's treated in the sarcoma community by a sarcoma specialist. And so if you're being treated by someone who treats all different types of cancers, and so sarcomas are 1%, leiomyosarcoma is then 0.2%, the ability to understand and pick up this and read this is less because they're focusing on breast cancer, colon cancer, lung cancer, and everything else walking in the door. So it's not easy to be up and current, but certainly as a patient, you advocate for yourself. If you're up and current, then you bring that information into the office with you. Okay, and here we have a small study of the safety and efficacy of a combination of lurbinectedin plus doxorubicin, and this is an early phase clinical trial. It is for advanced soft tissue sarcomas. And here again, what we see, the similar waterfall plots. So this 0% is where everybody starts. And then if the line goes down, that means their tumors get better. If the line goes up, that means the tumor gets worse. And what you see is the blue ones are all patients who respond. And if you're looking at the responders, This person here is a leiomyosarcoma. This person here is a uterine leiomyosarcoma. This is a uterine leiomyosarcoma. That's a uterine leiomyosarcoma. This one's hiding, I don't remember, sorry. But essentially, leiomyosarcoma, uterine leiomyosarcoma, the patients are doing well. And so this really is looking at a new therapy to see whether or not lurbinectedin could be used for our patients in combination with doxorubicin. And if this trial continues and it gets further, which it is currently being evaluated further, then essentially what you do is you want to look and see, is it better than this? And the numbers you're going to look at is, what did you achieve with doxorubicin and trabectedin? And if lurbinectedin is a better drug or equivalent, then you want to look, if it's an equivalent drug, is the side effect profile better? And so there's a lot that goes on when we do studies versus studies. If they're all being done within the same era, then it's very easy to play them against each other. If they're being done in ones 20 years ago, then there's these historical changes and everything has changed over time. But all these studies now are done in the same era, which makes us have the chance to really analyze, look at it, and come up with the best combination for patients. And the last thing I was going to talk a little bit about is precision medicine. And so what you have often, and when we look at the genetic landscape, is you often have, you could divide sarcomas into two main characters based on their genetic features. You could have ones that have a translocation or a fusion driven. The translocation means almost like two chromosomes break apart and they merge together, or like a piece of one goes to a piece of another. That's sort of a fusion. And then complex karyotypes, meaning that when you look at your report, there's a lot of changes. So sometimes you have amplification, meaning you have too much of something. Sometimes you have deletion, meaning something fell off. So there's certain changes that happen. The problem with leiomyosarcoma's genome is that often targetable alterations are, with the currently available therapeutics, aren't available. And so it's not common to have too many of these fusions where we have drugs that target the fusions. It's not common to have amplification or deletions of certain things that we know we have targetable drugs for. So when we look at the top line is the most common mutated genes. And again, we see this is soft tissue leiomyosarcoma, this is uterine leiomyosarcoma. We see the most common is going to be TP53 on both sides. We have the RB1, which I talked about before, the MED12, and you have the MED12 there, the RB1 there. There isn't a lot here in these mutated genes where we have drugs that will target it. The bottom part is when you have too many copies. And so it's amplification or alteration in copies. So it's AMP is amplification, meaning that you amplify, you have too much of something. And DEL is a deletion, meaning you don't have enough of something. And so even if you look at right across the board, this CDKN2A deletion, you potentially have a drug for that, which is a drug called palbociclib or abemaciclib. But that is really all that you have there. And so what we realize is either we have to have better testing to find things that we're missing, or ultimately we have to get smarter and start to target some of these things, because where we currently lie is lacking in targeted therapies for leiomyosarcoma. So there are no FDA-approved targeted therapies specifically for leiomyosarcoma. We do have pazopanib, or Votrient, which is FDA approved for advanced soft tissue sarcomas, including leiomyosarcoma, that received FDA approval and is good for first line, second line, or subsequent lines. Other than pazopanib, the only approved targeted therapies for leiomyosarcoma were those that had a tissue agnostic approval. So tissue agnostic means it doesn't matter what the tissue type is, it matters what the genetic profile is, and that every patient with the same genetic profile gets access to these drugs, whether it's a colon cancer, a leiomyosarcoma, a breast cancer, any other cancers. And so we know that immunotherapy with pembrolizumab or dostarlimab is approved if on your genetic test, your tumor mutational burden, so your TMB number, everyone has a TMB number on your pathology, on your genetic test, and that number needs to be above 10 in order for you to be eligible for one of these drugs, or you need to have a microsatellite instability high. Most of you will have MSS, which is stable. You don't want it to be stable, you want it to be high. The likelihood of finding those, I mean, it really is a needle in a haystack, unfortunately, but every once in a while we do find it, and these patients do very well with immunotherapy. If on your report you have a BRAF V600E mutation, then you get treated like melanoma. And I said before, I had a Leiomyosarcoma patient who got treated like melanoma because she had a BRAF V600E mutation. And then you get drugs, dabrafenib and trametinib, instead of getting the classic chemotherapy that we give most of our patients. If you have a RET fusion, you end up getting selpercatinib, which is approved for thyroid cancer. And then there are these drugs for, we talked about fusions, when two chromosomes fuse together. If one of those fusion involves NTRK, NTRK, then you're eligible for entrectinib or larotrectinib, which the drugs work in probably close to 100% of patients. But the NTRK fusions I see maybe once a year. So they're not common. So based on the GE database, 9.7% of soft tissue leiomyosarcomas and 6.5% of uterine leiomyosarcomas have a high tumor mutational burden. But if you don't do the test, you don't find this out. And this is why the importance of doing genetic testing is important. If your tumor sample is old, meaning more than 2 years old since you've had a surgery or biopsy, then you could do samples through blood, and that's what we call cell-free DNA or circulating tumor DNA. So it's either as a small c, small f DNA, or a small c and a small t. So circulating tumor or cell-free. And then with respect to looking at some big databases, only one uterine leiomyosarcoma case had a BRAF mutation, one had a RET fusion, one had an NTRK fusion. So they do exist, they're not that common, but we need to be looking for them because the therapies are highly, highly effective. And so here we have tumor mutational burdens. And so what we see is that a tumor mutational burden less than 2 is the most common thing we see in all of our sarcomas. We see it, about 1.7 is the average tumor mutational burden that we see. And so what we need is the high, which is above, usually above 10 we want, but above 16 certainly. And here we see that's a very small percent of patients. But certainly if you're one of those patients, then the treatment that we should be giving is immunotherapy. And so I added this, I think this is my last slide, and the reason I added this is just to stress how complex everything is. This is the thought process we all go through. And the thought process that we want to understand is essentially this is considered the inside of a cancer cell. That's the outside of the cancer cell. And so either you want to target things in the environment around the cancer cell, so you're either targeting a T cell, and so immunotherapy targets T cells. T cells will then go fight the cancer. You want to target the macrophages because you're going to somehow impact what's happening to the cell. Or you go and you attack something on the inside of the cell. And on the inside of the cell are all these pathways. It's like a recipe where one goes to the next, goes to the next. And so you want to see what you could target, what you could hit. And as you hit them, what you hope ultimately happens is that you hit something and makes it more sensitive to something else. And so that would make sense to do combinations. So if I knew, just talking off the top of my head, this isn't true, but if I knew that if I knocked out this, it makes you more sensitive to doxorubicin, well then it makes sense to do a combination of this with doxorubicin. And so it's sort of, we look at this to try to figure out how best to form a pathway. Could these work by themselves? So a lot of patients with breast cancer are treated with PARP inhibitors. There's a role for that in sarcoma as well. So we certainly have to understand the pathways and understand the drugs that we have available. And so that's the process when we're thinking about designing clinical trials, we're thinking about new drugs. Everything is based on understanding pathways, understanding why the cancer forms, why the cancer succeeds and survives, and so how to alter some of that. And I think that's basically it. It's probably, there's a few more certainly to thank the Leiomyosarcoma foundations for doing these symposiums, certainly helping patients. And understanding the basis of a lot of these advocacy groups and a lot of support groups and research groups are really to award funds to be able to provide research seed money so people could do the work that they want to do. It's to collaborate with other groups, and so if you're working with one group doesn't mean you can't work with the others. There's a lot of groups in the sarcoma space that all work together. So the focus is often on getting research done, and that's the part of the physicians and researchers and PhDs. The other part is really trying to understand and educate patients so that patients have the best knowledge, because you actually may carry more knowledge going into the doctor's office than your own physicians, depending on who you're seeing. And so that's the last slide. So together we will move forward and tackle our greatest challenges.
Right on.
Thank you, Dr. Agulnik. So we do have time for some questions. So yesterday we gathered together at dinner and we came up with some questions then, and also from those in the room that we might have some questions here too, based on your presentation. I did actually have one main question, which is based on your presentation, which is so the importance of doing the genetic testing. The genomic testing is really important. Is there a list in terms of the panel testing that we as patients should be going and armed with to say, can I have this, and this? You know, I'm from the UK. You know, I had to push even just to get NTRK testing, for example. So, for colleagues here, is there somewhere that they can go to find out actually a typical panel? What should I be trying to look for so that I can go? And also, you mentioned about the synovial sarcoma possibility for LMS. Actually, what mutation are you looking for that potentially would mean that an LMS patient would be able to be, you know, would be relevant for that synovial sarcoma treatment?
Thank you. So the goodness of the NGS test is it's very comprehensive. And so when the drugs first come to market, it's hard as a patient, because it's hard as a physician to really know, are you ordering the right test? Most of the tests will have a specific sarcoma panel, and you know that you could order that. But when you talk about the NTRK, because it's interesting, so I traditionally have used one of 3 different companies. And so there's NTRK 1, there's NTRK 2, there's NTRK 3. 2 of the companies did all 3, 1 company didn't do NTRK 2. So then it was very important to know I have to stop using that middle company. I don't think as a patient you could get that type of granularity. I think what you have to realize though is that these genetic testing are not live documents that continue to change over time. And so if you ended up having your genetic testing done today, and in 6 weeks a new drug came to market that has a specific role in what you have, it's not going to show up on your NGS test because your test is outdated already because it's 6 weeks old. They don't keep updating them based on the information. So what I would say for most patients is, so if you had a test done and then you have this very sweet period of several years where you don't need any active therapies and you're doing great, but then unfortunately something happens and you start to need new therapy, I believe you have to redo an NGS test because it's outdated, and you want to make sure that your current test has essentially is being tested for the right thing. Because if you ended up, so before NTRK came to market, these tests were not checking for NTRK. And they shouldn't have been, because there is no target for NTRK, and so it didn't make sense to do it. But then once a drug comes to market, it's very important that this is tested, because now you have access to something. And so if you had a testing done 2 years before we knew about NTRK, well then your report is not going to reflect that. And so do I think everybody has to go out and continue to have biopsies in order to do these NTRKs? Absolutely not. I think fresh tissue biopsy is wonderful if you could do it and if it's not going to cause harm. But I think you could often do these through blood. And a simple blood test certainly will give you the information you need. And for me, the time periods to do the blood test is going to be any period where there's a change. And so if you spend 2 years on a drug therapy, or if you were on the drug holiday for 2 years and then something changed, in my mind, the genetics changed, which is why your disease is different, and I need to know that information. And so I think that I would personally, if it was my body, trust any commercially available panel. I trust the panels when I see patients from UCSF and they have a panel, I trust their panel. When I see them from Memorial Sloan Kettering, I trust their panels. When I see them from City of Hope, I trust their panels. I trust the in-house panels because those are validated and they're all like CLIA lab approved, and so they are all high yield panels. But certainly we've been testing patients probably for the last decade plus, And we've seen changes, and companies come all the time. I just saw someone from Guardant yesterday, and she goes, oh, we have new panels. Like, so there's always, they're finding new things and they're testing, and so that becomes very relevant for people. With respect to the synovial sarcoma drug Tecelra, that is only for synovial sarcoma, but it was a matter of that the technology that we use to figure out that drug is technology that we could use to apply leiomyosarcoma. And so that specifically is looking at like the TP53. Synovial sarcoma is very different because it has a translocation, and it has too much expression of NY-ESO and too much MEG-A4, which isn't replicated in leiomyosarcoma. It's replicated sometimes in like some esophageal cancers, some certain ovarian cancers. And so further, like we have another trial that we're starting at USC for synovial sarcoma, for myxoid round cell liposarcoma, and then also for gastroesophageal tumors, ovarian, and it's because those could have a similar makeup.
Thank you very much. Then there was another question which was about, so you talked about pazopanib. How do people in this room know whether or not that's something that is something that is beneficial for them or not? What are the things that they should be looking out for?
So based on the data, and the data is a phase 3 clinical trial that used pazopanib versus placebo. It was a European study called the Palette study. Every patient was then given access to pazopanib if they were on placebo originally. And it was all soft tissue sarcomas except liposarcoma. And essentially the leiomyosarcoma group did just as well as every other group. So they teased it out for all soft tissue sarcomas, for leiomyosarcoma, and for synovial sarcoma, and all three groups did identical. And so it is an effective drug, and unless you have a contraindication to getting it, it probably should be used in every leiomyosarcoma patient at one point in the journey. The contraindications to it is that it suppresses the ability to heal a wound. And so if you have, let's say you have a sarcoma that's really in the skin, so I have someone it's really like in the skin by the ankle, and the wound never heals. Well, then this isn't a good drug for that person because the biggest thing they're suffering from is an open wound. So the last thing they want is to add a drug that's going to make that worse. It's not a good drug if you think someone's going to have a surgical procedure soon. You also have to look at, if you're really prone to nausea, or if you have a condition of a long nausea, you can't take Zofran with it, or ondansetron. You have to look at other things, but for the most part, this is a good drug for patients.
Thank you very much. So the tables yesterday had asked, how can we promote collaboration or share best practice in terms of research, particularly internationally as well?
I think within the sarcoma community, we do a very good job. I think for those of you in the LA market, you've probably seen many of us. There's a few of us in the LA market doing sarcoma, and then every time you come to a consult where you want to get a second opinion from one of us, probably what we say is, oh, we know the other person, we're friends with the other person, and we're texting the other person. So the community is very, very close-knit. There aren't that many sarcoma medical oncologists throughout the United States. So LA market is a very resource-rich market because you could go to UCLA, you could go to Santa Monica, you could come to USC, you could go to City of Hope. There's many places you could go. If you're in Nebraska, maybe you're a little bit more of a vacuum. The Las Vegas market's a difficult market in Nevada. And so I do think that here it's a very resource-rich market, and I think the sentiment I get from most people when they get second and third opinions is that the opinions are very similar because we all share very much the same information. We're all doing similar trials at different times. We all go to the same meetings. And then it becomes an issue of personality and location. And so you do, in this market, do have the opportunity to be treated a little bit closer to home, depending on where you live, because there's quite a few sarcoma specialists. When I first got, I worked for 14 years in Chicago at Northwestern University, and I was the only one in the city. There was someone doing sarcoma work in the suburbs. It's changed over time, and different universities did hire, but then it became a little bit more of a vacuum for people and a little bit harder. So if your insurance allows you to be seen at a academic center with a sarcoma specialist, that's probably in your best interest, but not everybody. Some people have HMOs that don't allow it. I mean, you have to do what you could do. You can't go bankrupt because you want to pay out of pocket. It just isn't reasonable. But I would say that collaboration within the US is phenomenal. I'd say collaboration overseas is phenomenal, certainly through Europe. Europe has a very strong sarcoma network. It's very different in Europe, it's different in Canada. So you can't, whereas here, like we were talking about several hospitals all having sarcoma specialists, things in Europe and things in Canada with socialized medicine ends up more centralized. And so it's easier to have 5 sarcoma doctors in one space, because that's better for sarcoma, rather than 5 sarcoma doctors in 5 different spaces. And so the burden ends up being that the patients have to travel too, but the care is better. Whereas here, everybody wants to be close to home, which totally makes sense. And so it becomes one of those things where you have several of us, and the way we collaborate is through meetings, tumor boards. Everyone has everyone's cell phone number, and everyone's texting each other all the time. We're texting because we want to see, does someone have a clinical trial? And so if I don't have something at USC, I want to know, does UCLA have it? Because that's better for the patient. And so we are all shifting people around based on the availability of different trials.
Thank you. I think we had someone in the audience who had a question, but then I think we probably have to carry on with the next.
Yeah, thank you, Robert. For some uncommon gene mutations like KIT, NOTCH2 and JAK3, these are uncommon mutations. Were there any clinical trials in immunotherapy which have failed, probably because the drug was not effective? Or is it because those gene mutations are uncommon and that's why there are not enough clinical trials?
So the question, if I'm understanding correctly, is how do we address rare mutations for patients? And so there's huge initiatives done through all of oncology and cancer care. And so there were huge initiatives done from very large cooperative groups where it's most of the academic centers and a lot of private practice centers throughout the United States. And they would be like, there was a match trial. So the match trial would try to match people based on their genetic profile, irrespective of the tumor type. And so it wasn't a leiomyosarcoma trial, but like when I talk about my patients with being treated with other drugs, they were on the match trial because the match trial was the easiest way to get access to drugs for patients, and then the drugs are free for the patients. And so I had a patient years ago with a very high tumor mutational burden. That was part of, that's how we know that these drugs work in high mutational burden, because they were part of the match trial. And so they got atezolizumab on that trial. Are there trials in immunotherapy that have not succeeded in this space? And so the reason that a lot of patients in this space are not offered immunotherapy is because of the trials that haven't succeeded. And so there were trials specifically in uterine leiomyosarcoma that were closed very early because within the first 8 patients, not a single person got a manifest. And it probably is because these tumors are not immune responsive tumors. However, our understanding is different. Sometimes when you give a lot of treatments, the tumors then increase their tumor mutational burden because the tumors get angrier the more things you give at them, and then they may be responsive to immunotherapy. And so whereas like if you have a melanoma, immunotherapy plays a role right away, in sarcoma it probably has a much more delayed area for leiomyosarcoma. There is only one sarcoma, the alveolar soft part sarcoma, that does have an FDA approval with atezolizumab, because that is the only sarcoma that highly responds to immunotherapy.
Let's just have one more question.
Perhaps this is just a practical question. We've been through the experience with United Healthcare rejecting any of the costs for these tests that you referenced from 3 different institutions, and the doctors don't seem to have a way to address the appeal to make it go through. Have you had any experience, or do you have any guidance to offer how we can have more success?
The testing you're asking about is NGS testing. You're asking about testing, like genetic testing on the tumor by other companies.
Correct.
Okay, so my understanding, and I haven't spoken to one of them in a little bit still, my understanding is that if they did not recuperate the cost from the insurance company, that they don't push it further. They don't take patients to collections, is my understanding. And so there's a lot of debate on what should be covered, what shouldn't be covered. A lot of these drugs need a NGS report in order to get approved. And so like an NTRK fusion, you could only find it if you did a genetic testing. The company specifically did not tie their approval of the drug to the approval of an NGS because they wanted patients to be able to get any test and not just one. But my understanding from, I've never had a patient report to me that the company has taken them, that is, I think they send them 3 or 4 letters and then it is stopped, is what I've been always told. If that is changing now, that would be very unfortunate because these are very expensive tests. They could be in excess of $9,000. And so, but my understanding is they push the insurance, they push the insurance, they push the insurance, and then it stops. I don't know if your experience is different.
We never got any results, and they never produced the test.
They just tell you what drug to take at the hospital, which should they give.
And the insurance company said that
We could
there's
talk
no benefit.
privately, but there's a disconnect there. Because usually these tests are done and then they're billed for afterwards. Because you don't bill for a test if the test, so not every time you send a sample, you may not have a good sample, and so not every test generates something, and so they would never pre-bill someone.
Does Count Me In cover the NGS? Does Count Me In cover?
I don't know.
You don't know. Okay.
So for people who are here and for the general community, is it worth individuals, if their insurance company is not willing to pay for it, is it worth them putting their hand in their pocket or saving the money to get an NGS test done with the full panel and then come to you to say, hey, this is what it looks like? Would that be helpful? And would that be in their best interest?
So I think in an international patient group, I think you are stuck, and that would be yes is the answer. So I think if you were in England, if you were in Canada, and these things are not available, the same reason that patients would go and pay out of pocket for mammograms that aren't necessarily scheduled at the intervals that we like them to be, we're passing it. So, but in an American system, we're paying so much already for healthcare, and you're paying for everything. I don't think that this is something else that you should have to pay for. This should be part of standard of care. And my understanding is that patients will get bills, but they don't push it to collections. And so these companies, if you've gone to collections, I'm really sorry. But it's always been my understanding that the companies have always told us that they will continue to do these tests because they probably make enough money from all the insurance companies that do cover them already.
Okay, I really think we really need to carry on, but he's waited so patiently, I feel.
Last one.
How good is cell-free DNA as a diagnostic?
So not as a diagnostic. A diagnostic to me is the ability to diagnose a problem. So cell-free DNA will not diagnose a problem unless...
Well, a doctor will diagnose mutations. How good is it in diagnosing mutations?
Yeah, so it's not, so cell-free DNA is a blood test. It's not as good as tissue because you may not have circulating tumor cells. But if you don't have tissue available, it is a very reasonable first place to go because everyone knows, most people have had a biopsy. A blood sample is a lot easier than a biopsy. And the turnaround time for cell-free DNA is about 7 to 9 days, depending on some of them faster. The turnaround time for tissue could be 6 weeks. And so if you want to see something quickly, you could do cell-free DNA. And then if you get nothing back, then you could discuss doing the biopsy, and all you've lost then is a week. And so you just have to sequence it and put it into, sometimes you have to be practical. And so for me, I send off blood all the time. Because if I capture something, then it negates the need for someone to go for a biopsy.
Okay, so the very, very last question is that we've been asking all the speakers so far is, if you had an unlimited pot of money towards LMS research, what is it that you would use that money on?
Unlimited. So I think tissue banking. And so often universities will bank tissue that they could use later for research. I think that's very important because as evolution happens and we get more information, you don't want to be stuck having to say, oh, I think this is very important, so now let's spend the next 5 years collecting tissue and then we can work on it. Instead, you could go to the bank, unfreeze the tissue, and you have 5 years of tissue, and I think that's the fastest way to get things along. I think if I could The NGS, so the problem with the NGS reports for most places is that it's an external company who does it, and they don't live in an electronic medical record. And so the problem is they're very hard to find. So if someone comes to me for a second opinion, I often say, did you have NGS testing? Often they say yes, often I say what company, they don't know. They don't know the results necessarily, and that's normal because you have so much information that you have to be the bearer of. And then a lot of people say, oh, it's in my record. But if you go into your Epic, and a lot of people have Epic, the NGS report is not in Epic because the Epic is everything that was done. Let's say you're at UCLA. UCLA, everything done at UCLA is in Epic. If you had it done through Foundation Medicine or Tempus or Guardant or Caris, it's in that portal. And so because the portals don't link, I would use, if I had unlimited funds, you'd want to make sure that everyone's NGS report lives within Epic or within Cerner or in the electronic medical record so that the information could be used more freely.
Great, thank you so much, Dr. Agulnik. That's been absolutely fantastic. Can we also say thank you? Right, great. So the next, so the next presentation, I'm just going to stop the recording.
Recording stopped.
And actually, we also wanted to have a big group picture. So if it's possible, I don't know, Dante, is it possible for us to try and